Hi, it's Ms. Vitale. This podcast is on bacteria and archaea, and it is meant to correspond with Chapter 27 for AP Bio. Bacteria and archaea are the smallest organisms. They can be 0.5 to 5.0 micrometers, which is 5% the size of most eukaryotic cells. They are the most numerous organisms on Earth. There are more prokaryotes living on and in our bodies than we have human cells. Like eukaryotes, prokaryotes conduct glycolysis, use DNA to encode proteins, do transcription and translation with the same genetic code, replicate DNA semi-conservatively, and have plasma membranes and ribosomes. However, they differ in three important ways. First, they lack a cytoskeleton and nucleus, and they divide by binary fission, not mitosis. Second, DNA is usually circular and not divided into chromosomes. Bacteria DNA also lacked histones and introns. And third, there are no organelles except for ribosomes. Although prokaryotes are single-cell, they often like to form colonies. Most prokaryotes have a cell wall, in particular bacteria. Not all archaea have cell walls. The cell wall gives them shape and protects them. In bacteria, it is made of peptidoglycan, which is a complex of sugar and proteins. Archaea cell walls do not have peptidoglycan. Gram staining is used to classify bacteria. It was developed by Hans Christian Gram in the 1800s. He was a Danish doctor. First, it exposes the cells to crystal violet, then iodine, then they're rinsed in alcohol, and then they're exposed to a red dye. If they are gram-positive, they stain purple. Gram-positive bacteria have a simple peptidoglycan cell wall, and it takes up a lot of stain. They tend to be less dangerous because they can easily be attacked by the immune system, and antibiotics usually kill them readily. Gram-negative are thinner peptidoglycan cell walls with an extra carbohydrate lipid membrane. Oftentimes that membrane contains toxins, and that membrane also protects it from the body's defenses. Gram-negative bacteria tend to be more dangerous, and they resist antibiotics. We can also classify bacteria by shape. A round or spherical bacteria is considered a cocci or coccus. A rod-shaped bacteria is bacilli or bacillus, and a spiral-shaped or corkscrew can be a spirochete or spirilla. The prefixes for describing how bacteria clump together: mono means one, diplo means two, strepto forms chains, and staphylo forms clusters. For example, Streptococcus is a chain of spherical bacteria, and that is the type of bacteria that can cause strep throat. Other structures are found in some bacteria. Some bacteria have a capsule. This is a sticky layer that helps it to adhere to substances or to each other. Fimbriae, which are also oftentimes called pili, or the single form is fimbriae, they are short projections. They are also for sticking to things and for sticking to each other. Some bacteria can move. Some bacteria use a flagellum to move. Flagella are slender filaments that extend singly or in tufts from one or both ends of the bacteria cell, or they can be distributed all around the cell. They are not the same as eukaryotic flagella because they're only about 1/10 the width. They're not covered with the plasma membrane. They're not made of microtubules. Instead, they're made of flagellin, also a globular protein. They have no basal body at the base, but their basal apparatus is made of rings embedded in the cell wall. Spirochetes are helical bacteria that use a corkscrew motion. That's made possible by modified flagella that run beneath the outer membrane. Many cyanobacteria and a few other groups of bacteria use poorly understood gliding mechanisms, including rolling. Some move up and down in the water by adjusting the amount of gas in gas vesicles. The term taxis is the movement towards or away from a stimulus. Positive chemotaxis is moving towards nutrients. Negative chemotaxis could possibly be moving away from toxins. If we look at the genetics of bacteria, most bacteria have a circular loop of DNA with no proteins and fewer genes than eukaryotes. They have about 1/1000th DNA of eukaryotes, but about 100 times more than viruses. The nucleoid is their packed area of DNA, and it has no membrane about it. It's also usually a continuous loop. Bacteria asexually divide by binary fission. The loop of DNA forms replication forks. It eventually copies the entire circle, and the two chromosomes move into opposite sides of the cell. The cell begins to expand, divide in the middle. The cross walls eventually divide the two, and you wind up with two identical daughter cells that were clones of the original parent cell. Bacteria can reproduce very quickly in the right environment. For example, E. coli reproduces every 20 minutes. Since binary fission is asexual and produces offspring identical to the parents, you would expect there not to be mutations, but mutations can create different offspring. About 1 in every 10 million cell divisions has a mutation. If the new mutations are favorable, that cell will reproduce itself and proliferate. This is one of the ways that bacteria become so genetically diverse, and because they multiply so rapidly, 1 in 10 million is not as small of a chance as you would think. Bacteria can also exchange genetic material between two cells in 3 ways. The first is transformation. Transformation, if you recall, was discovered by Griffith. A bacteria cell incorporates DNA into its nucleoid by taking it in from the surrounding environment. In Griffith's experiment, a live bacteria took DNA from a dead one. When that happens, recombination occurs. Recombination is the exchange of DNA from 2 different sources. Transduction is when a bacteriophage, or a virus that infects bacteria, takes bacterial genes from one host cell to another. The phage attaches to the cell, injects its DNA. The DNA of the phage breaks up the DNA of the bacteria. The bacteria is forced to make new viruses, but instead of a virus forming with the original viral DNA, it inadvertently picks up a piece of the bacterial DNA. So when that virus moves to a new bacteria cell, it will inject a piece of the previous bacterial cell into the second one. The other bacteria's DNA becomes part of the new bacteria's DNA, and you wind up with a recombinant bacteria. Conjugation is bacteria sex. 2 bacteria come together and exchange DNA. The cell that forms the pilus, which is an extension, is considered the male. That male cell donates DNA to the other cell, which is considered the female. The male cell forms the pilus, attaches it to the female, and pulls her towards it. A cytoplasm bridge forms between the two, and there is exchange of DNA, usually in the form of plasmids. The male bacteria has the F factor, which stands for fertility. This is a section of DNA, or a plasmid, that enables it to form the pilus and donate DNA to the other cell. Recall that plasmids are circular pieces of DNA that self-replicate in bacteria cells. Episomes are DNA that can form a plasmid or become part of the bacteria DNA. In other words, they're sections of DNA that can enter the larger chromosome, the nucleoid, or leave and become a plasmid. As I said before, the F plasmid is for forming a pilus, and a cell that has the F plasmid is considered F positive, and it is a male. F negative is the cell without the plasmid and is considered female. R plasmids are plasmids with genes that make them antibiotic resistant or resistant to something else like disease or other pests. Some bacteria can survive harsh conditions by forming endospores. Endospore is when the cell produces a copy of its DNA and surrounds it with a tough wall. They can only be killed by an autoclave. That is a piece of equipment that heats under pressure to 121 degrees Celsius. Boiling, which is 100 degrees Celsius, will not kill endospores. They have been found in mummies thousands of years old. Bacteria do have ways of communicating. They can send and receive messages. Bacteria can release chemical substances that are sensed by other bacteria of the same species. For example, they can announce their availability for conjugation, or they can monitor the density of their population. This density sensing technique is called quorum sensing. Like fireflies and many other organisms, some bacteria can emit light by a process called bioluminescence. This is a complex enzyme-catalyzed reaction that requires ATP and causes the emission of light, but not heat. Prokaryotes have amazingly diverse metabolic pathways. They outdo eukaryotes in terms of metabolic diversity. Some prokaryotes can live only by anaerobic metabolism because molecular oxygen is poisonous to them. They are called obligate anaerobes. Other prokaryotes can shift their metabolism between aerobic and anaerobic modes. They are called facultative anaerobes. And obligate anaerobes are unable to survive in the absence of oxygen, so they require oxygen. All living organisms face the same nutritional challenges. They must synthesize energy-rich compounds like ATP to power their metabolism, and they must obtain carbon to build organic molecules. Biologists recognize 4 broad nutritional categories for organisms. Prokaryotes are represented in all 4 of those categories. The first one is photoautotrophs. They perform photosynthesis. Their energy source is light and their carbon source is CO2. Cyanobacteria use chlorophyll A as their key photosynthetic pigment. Photoheterotrophs use light as their energy source but must obtain carbon from organic compounds made by other organisms. For example, compounds released by plant roots, like in rice paddies, or from decomposing photosynthetic bacteria in hot springs, are taken up by photoheterotrophs and metabolized to form the building blocks for other compounds. Sunlight provides the necessary ATP through photophosphorylation. CHEMOAUTOTROPHs obtain their energy by oxidizing inorganic substances, and they use some of that energy to fix carbon dioxide. For example, deep-sea hydrothermal vents are ecosystems that are dependent on chemoautotrophic bacteria. They're the basis of the food chains in these communities at depths of up to 2,500 meters. This is below any hint of sunlight. They get energy by oxidizing hydrogen sulfide, released in the near boiling water flowing from volcanic vents in the ocean floor. Chemoheterotrophs are consumers who get energy and carbon from organic compounds produced by other organisms. These are your normal heterotrophs, and chemoheterotrophs also include all animals and fungi. This is an overall table of the 4 different types of nutritional categories. Many important reactions in prokaryotes involve nitrogen or sulfur. Some use nitrates, nitrites, or sulfates as electron acceptors in their electron transport chains instead of oxygen in cellular respiration. Denitrifiers are bacteria that release nitrogen to the atmosphere as nitrogen gas. For example, the genus Bacillus, which includes anthrax, and the genus Pseudomonas, some of these are human pathogens that cause infections, use nitrate, which is NO3-, as an electron acceptor in place of oxygen. Nitrogen fixers convert atmospheric nitrogen gas into a usable form, usually NH3, which is ammonia. These bacteria are found in the soil or in the roots of legumes. Legumes include peas, beans, soys, and alfalfa. All organisms require nitrogen to build proteins, so nitrogen fixation is vital to life as we know it, and these prokaryotes are the only organisms that can do it. So nitrogen fixation is an extremely important process, and that cannot be underemphasized. Nitrifiers are bacteria which are chemoautotrophs that oxidize ammonia into nitrates, in turn using the energy released by this process. In some cases, a few specialized cells in a colony called heterocytes carry out a function different from the rest of the colony. For example, there are some photosynthetic cyanobacteria that have genes for nitrogen fixation in addition to photosynthesis. But they both can't happen in the same cell at the same time because oxygen produced by photosynthesis prevents nitrogen fixation. So instead, most of the cells do photosynthesis, but there are a few occasional cells that are separate and they carry out nitrogen fixation and not photosynthesis. Those are the heterocytes. When looking back at the evolutionary history of prokaryotes, some genes have moved sideways from one prokaryote species to another. This is called lateral gene transfer. Although this usually occurs between related species, some have been documented across the three domains, ARCHAEA, bacteria, and eukaryotes. This makes classifying prokaryotes complicated. In addition, the great majority of prokaryote species have never been studied. However, we can still divide prokaryotes into the two main groups of bacteria and ARCHAEA. Let's start with ARCHAEA. Originally, all ARCHAEA were thought to be extremophiles. That means that they're able to survive in very extreme environments, oftentimes reminiscent of early Earth. Extremophiles include halophiles, which love salty water. We find them in places like the Dead Sea and the Great Salt Lake, which have a very high salt concentration. Extremophiles also include thermophiles. They love very hot environments. These are the types that you find near the deep sea hydrothermal vents and also in hot springs. It turns out not all ARCHAEA are extremophiles. More have been discovered in moderate environments. METHANOGENS are strict anaerobes that produce methane. We find them in marshes and in the guts of herbivores. They are also used in sewage treatment. Spirochetes are gram-negative, motile, chemoheterotrophic bacteria. They have modified flagella in their plasma membrane, causing the cell to rotate. They can live free in mud or water, but they can also be human pathogens. Some of them cause Lyme disease and syphilis. Chlamydias are very small, 0.2 to 1.5 micrometers. All of them are parasitic, and they are gram-negative cocci. They cause eye infections, which is the most common form of blindness in the world, and they cause the most common STD in the United States. High GC gram-positive bacteria, also called actinobacteria, have a high ratio of guanine cytosine base pairs compared to AT or adenine thymine base pairs. Many are a valuable source of antibiotics. For example, Streptomycetes produces streptomycin, a common antibiotic. However, tuberculosis is caused by a high GC called Mycobacterium tuberculosis. Cyanobacteria, which we've already talked about, are important photoautotrophs. They only require water, nitrogen gas, oxygen, and a few minerals. They were the basis for the oxygen revolution. Some can differentiate into vegetative cells, which do photosynthesis, or into spores, which are resting stages during harsh conditions, or into heterocytes, which perform nitrogen fixation. Low GC gram-positives have a lower G to C and T to A to T ratio. They are capable of producing endospores. Bacillus anthrax that causes anthrax is an example. The anthrax spores can live in decomposing organisms and in the soil for centuries. Staphylococcus aureus is the best known human pathogen and is found in 20 to 40% of normal adults and 50 to 70% of hospital adults. This is the cause of staph infections. Mycoplasms, which are in this group, are the smallest of cells. They are below 0.2 micrometers. Proteobacteria are a large and diverse group. They are all gram-negative. Genetic evidence suggests mitochondria evolved from a proteobacterium. There are 5 groups of proteobacteria based on different metabolic pathways. Their common ancestor was a photoautotroph. Early in their evolution, 2 groups lost their ability to photosynthesize. The other 3 groups have some photosynthetic members and some that have taken up other modes of nutrition. Alpha proteobacteria include many that live closely with their hosts. Rhizobium are nitrogen fixers that live in the roots of legumes. Beta proteobacteria include many bacteria strains that contribute to the nitrogen cycle. Gamma proteobacteria contain some bacteria that cause disease, like Salmonella food poisoning and E. coli food poisoning. But remember, E. coli is also a non-pathogenic bacteria found in intestines. Delta proteobacteria include some that form fruiting bodies from their colonies. Fruiting bodies rise up like little lollipops and release spores. Epsilon proteobacteria are mostly pathogens. Helicobacter pylori is the cause of ulcers. This is a really interesting story. In 1982, a doctor who was treating a lot of ulcers named Barry Marshall discovered that when he took biopsies from most of his ulcer patients, there was a high level of Helicobacter pylori, and much higher than in normal people. He recruited Robin Warren, another doctor, and they started doing research together showing that in mice that ulcers are not caused by smoking and acidic food and drinking and stress, but in fact by a higher than normal level of these bacteria. When they presented their findings to other doctors in the field, they basically were ignored. The doctors did not believe them. So in order to prove that their theory was right, instead of testing on mice, they were not given permission to test on humans. So Barry Marshall took from a biopsy from one of his patients some Helicobacter pylori, mixed them in a broth, drank him, and created an ulcer, and then he cured himself with antibiotics. These 2 men eventually won the Nobel Prize for their work. So how do prokaryotes affect their environment? First of all, they are important players in element recycling. Bacteria, along with fungi, are major decomposers. If dead organisms were not decomposed, the earth would be covered miles high in debris. Many bacteria live in symbiosis with other organisms. There are 3 types of symbiosis. Mutualism is when the host and the symbiont benefit. For example, the bacteria that live in intestines. Commensalism is when one of the 2 benefits and the other is unaffected. For example, we have bacteria that live on our skin. They consume dead skin cells and oil secreted by our oil glands. We are not affected by them. And the 3rd type of symbiosis is parasitism. Parasites benefit and the host is harmed. If the parasite causes a disease, it is called a pathogen. Again, Mycobacterium tuberculosis, which causes tuberculosis, is an example of a bacterial pathogen. Bacteria cause disease by making toxins, unlike viruses that enter and destroy the cells. An exotoxin is secreted by the cell, and the bacteria doesn't even have to be present, just the toxin, to get sick. Clostridium botulinum is an example. This causes the very deadly food poison botulism. 1 teaspoon of botulism toxin can kill 1 million people. It is usually found in food that has spoiled, especially canned foods. Endotoxins are contained in the outer membrane of the bacteria. You must ingest the bacteria in order to get sick. And Salmonella food poisoning, which we often get from dairy products, is another example. Bacteria are used a lot in technology. We use them to make cheese and yogurt. E. coli are used to clone cells, which we've studied in the past. Agrobacterium are used to create transgenic plants, for example, creating golden rice, which contains vitamin A. Bioremediation is when we use bacteria to clean up environmental messes, like oil spills, and they are used to treat sewage in sewage treatment plants. Bacteria can also be used to synthesize natural plastics that break down faster in landfills, and we use them to create a variety of products, including proteins, vitamins, hormones, and antibiotics.